Everyone. I'll also give a timer for myself. I know I have a lot of information, so I will kind of speed up. Hi, everyone. My name is Xiang. I'm a computer science PhD student at the University of Minnesota, advised by Dr. Chad Myers. And today I'm going to introduce you a deep learning approach for predicting synthetic lethality in human cells. And let's start with defining what genetic interaction means. Genetic interaction occurs when mutations in two or more genes combine to generate an unexpected phenotype. So people really care more about this negative interaction shown on the left-hand side, which is, I'll give you a talk example. It's like in a healthy cell, if you knock out gene A, it's okay. If you knock out gene B, it's doing fine. If you knock out both, it suddenly dies. That's an extreme case we call synthetic lethal, which is a very ideal way of identifying cancer therapeutic targets. Successful examples are like PARP1 inhibitor and breast cancers. It's like if you identify an interaction partner that's only killing the cancer cells with certain mutations, but not killing the normal cells, that would be a very cool thing to see. And we do have a definition of positive interactions where it's really just like double mutations is not causing something as bad as you would expect quantitatively. So about a decade ago, our lab and our collaborators completed almost all the double mutants in the Baker disease, Saccharomyces cerevisiae, and from there, they were able to create a genetic interaction network with distinct functional neighborhood shown there. And the key rationale behind is that these interaction profiles are predictive of gene functions. In this toy example, like if you have a matrix quantifying gene-gene interactions, the genes showing high correlation across these profiles are actually assumed to be functionally related, more functionally related. And we can take that to scale up to create a network which we call profile similarity network, where the nodes are genes and the edges are actually the similarity, for example, Pearson correlation coefficients in their interaction profiles. So in recent years, with the emergence of CRISPR-Cas9 technology, we are moving from yeast to human cells. And thanks to our collaborators, we were able to use this TKOV3 lentiviral library to do this whole experiment. And we focus on the specific cell type HAP1, which is female-derived and haploid status cell line. And by comparing the single mutant fitness and double mutant fitness through a very complicated data normalization process, we were able to quantify and give a quantitative gene interaction scores to our pairs. And from there, we define negative and positive interactions. So I don't have time for these details, but I just want to mention this is like taking our previous postdoc, Max, several years of efforts to really trying to normalize and denoise from the raw reads to this final quantitative gene GI score with effect size and statistical significance. And our downstream analysis actually started from this matrix, which is a very valuable data set that we are publishing soon. On one hand side, it's almost genome-wide coverage of all the protein coding genes. And on the other side, we are adding one query mutant at a time. And now we have about 300 replicates. We have 300 query screens. And with that, we have 5 million double mutants created. So we were able to define some statistical cutoffs to really binarize to define negative and positive interactions. And if we really just care about the unique pairs after filtering the replicates, we were able to provide the community a data set of 4 million unique gene pairs, where about slightly more than 1% of them are defined as negative GIs and positive GIs. And there are 6,900 very interesting synthetic lethal pairs that I think will be very valuable resources. And similar to my toy example, if you take a snapshot of this giant matrix, and we can see that in this data-driven way, we can actually identify very clear clusters of functionally related genes. For example, if you look at the library side, vertically, you can see these FANG genes with known functions are clustered together based on their interaction profiles. And what's more valuable is like, if this is genome-wide coverage. So if you have an uncharacterized gene somewhere, the neighborhood information can really be very valuable in characterizing its functions. And on the query side, because we have some replicates for each query, we were able to show they are highly reproducible with high consistency and high quality in terms of the scores. So this, I would just briefly mention this unpublished map. It's like about 4,000 genes with high confidence relationships. And we were able to, just like we did in yeast, show distinct functional enrichment neighborhood there, which will be a very good reference map, because even just from these 4,000 genes, we were able to characterize not characterize, identify more than 100 uncharacterized genes there, and using this neighborhood information to help facilitate the process of the downstream studies. So this is the data sets. This is not even the downstream analysis, but we really want to mention this, and probably in the next month we will have a bioarchive about this big work. But as a computational scientist, the question I ask is like, when can we finish screening all of these pairs? Because we are doing this filling this little column one at a time, and let's see how other people are doing. Here is like in 2018, there's a big paper by Horvath et al. They constructed double mutants in 500 by 500 genes in 2 cell lines, K562 and JERCAD, and that's a cell paper. And we are doing we have many years of efforts as well, and we are having 4 million unique pairs here. But the whole space, if you just simply check, it will be 200 millions if you really account for all the possible combinations. So unless there's a breakthrough in biotechnology, I won't see that complete in my lifetime. So I have to figure something out in a computational way. So we identify a very good resource called DatMap by Broad Institute. They are doing some similar functional genomic screens, but they are doing doing single gene knockout across diverse cancer cell line backgrounds. So, and what we do is like pairwise knockouts. So these two are complementary data sets, and our last paper showed they are showing complementary signals. And what I really care about is like, if I can find association between our two data sets, and more specifically, I want to use this existing public data sets, big data sets, to predict our on-screen genetic interactions. So I kind of reframed this problem into a machine learning problem with pairwise units. For example, here, our single sample is no longer one gene, or one sample is like a unique pair of genes, so that even in my training data sets, I have 4 million samples. So we do have the gene interaction scores, and also we can binarize into labels to make a classification problem. And the key challenge is like, where are these features specific to gene pairs coming from? And yeah, and this setup is pretty, I'm happy with that, because if that map is like genome-wide coverage and I'm able to engineer pairwise features, I can have the features for all the possible combinations of genes, so that if I have a powerful model, I can really predict the rest of all on-screen GIs without filtering anyone. So yeah, that's the ambition. So before I show you some concrete results, I just want to mention the significance of doing this thing is like, yeah, we can infer the on-screen stuff, and using our prediction, we can guide and improve the experimental design. And what's really good is that our collaborators they keep doing this new screens, so that these are ideal experimental validation data to prove our model are good. And challenges here is like, yeah, where do we find pairwise features? Our labels are sparse, just as you see the gene interactions is really sparse. And we can also try exploring more resources than just that map, like TCGA or any embedded multi-omics data to be our pairwise features. So yeah, so we started exploring, and my colleagues Ke-Ching Chen here is exploring the more traditional machine learning ways, like attribute model. She had a poster here for her project, and we started there. It's like we can manually figure out these features that we think make sense to predict gene interaction. For example, co-expression, co-essentiality, expression dependency correlation, those kind of things that we human can think about. It's just 9 features there showing good performance, while I explore more on deep learning side, creating high-dimensional embedded vector from that map. And currently in the field, people are doing a lot of amazing things, graph neural networks, knowledge-guided methods, and they are borrowing representations from Go, PPI, knowledge graphs. I think they're awesome, but I always have this question, is like, if those are biasly annotated data sets, if you're going to predict something uncharacterized before, how are you going to generalize? So that's always my question, and that's why I chose to do a totally data-driven way instead of borrowing anything from Go or PPI. So I also want to mention a very important concept that this field sometimes overlooked. Is like, if you focus on predicting pairwise, a pair of genes, the data splitting strategies are very, you have to be careful about it, because if you still do the same random split of gene pairs, there could be a data leakage issue, or sometimes your model is over optimistic because they have seen this gene in the training set. Even it's not the same pair, it will still make the performance better than what its generalizability is. So in our case, because we are adding a query at a time, we are more like this cross-validation 2 scenario. It's like we only hold out query genes. We can train on the white space and predict on the orange space. While if you really want a tough setup, it can be CV3. It's like you won't see this gene, any gene of the pair in your training set and trying to predict them in the test set. So yeah, this is a very detailed thing, but I do want to share with the community. So yeah, how do I set up in a deep learning way? It's like I create an embedded vector for these pairs of genes. So firstly, I started with either the DepMap CRISPR knockout gene effect dataset and expression dataset. They have a similar setup, like a gene by cell lines. And I use autoencoder to compress this space, which I proved did not lose any predictive signals. And in this latent space, I took this vector of gene A and B, if I have a pair of them, to literally just add them up after correcting for their background and make it a vector make it a feature vector for me. So it's not a very complicated setup, but I took a version of this from the knockout data sets. I took another version of this from the expression data set so that I have 256 dimension of vectors in total as my features, and my labels are these synthetic lethal labels, which is pretty sparse. It's less than 1% of data set. And the neural network side is also fully connected neural network setup with 3 hidden layers. There are some hyperparameters here that we always want to optimize against, so I won't go into details, but actually I want to show even this kind of setup is good enough to show good performance. You don't have to go to a very large, complicated model. And one more slide before I show you the real results is like, what can we call as a good performance? So we literally took our biological experimental replicates as a reference. So for example, as I mentioned before, we have this multiple FANG-G query replicates, right? So we can take one of them and treat them as a predicted probability to classify the other replicates and see what's the kind of a ceiling performance can be. And we have this on the right-hand side, this benchmark evaluations. Like if you are using the real biological replicates to predict the other one, you can have a median AURC of 0.83 as a reference. You can do slightly better or less than this, but if you are far from this, something might be off. So we were able to show that, yeah, let's look at the left-hand side first. It's like if you have AURC in the easiest way, CV1 random splits, you have even better performance than that. And if you switch to a harder scenario, it's like drop a little bit. And actually boost is no worse than this in this setup. But if you look at AUPR, which I want to mention, PR curves are different because your background precision is kind of a reference point here, unlike the AURC. So it's like, yeah, you can see how random splits can be over optimistic. And if you switch to a harder scenario, the performance will drop a bit, although it's still more than 10-folds of the random background. And we mentioned this experimental validation data available, and I just want to give you one example. So we take one new query, this mTOR gene, as a holdout test, and we score it, and we use our model to predict on it, and we have an AURC of more than 0.87, which is pretty good. And it's like if you can figure out your model can predict something well and use that to guide these experiments, we can find this many mTOR interactions where only 2 of them, these are synthetic lethal interactions, so only 2 of them are shown with evidence in current literatures, which means actually the rest are even more interesting because we don't know much about them, and they're experimentally shown as synthetic lethal pairs with high significance. So yeah, so we worked on our own HAP1 data sets quite a bit, but we found out, yeah, if I'm using that map to create my features, actually my method can just span to all the data sets there because I don't have a limitation on our HAP1 data set. So we took this external validation data set from Holbrook 2018, and this time we can actually do CV123 to really finish the comparison. And the key message in this plot is like if we train even on 4 million of data sets on HAP1 and predict on K562 cell line, the performance is actually not good. It's like the cell line context is still very important, and this 0.55 AURC there. And if we really just train and test on the smaller K562 cell line, you can actually have higher performance even in the hardest double holdout scenario. It's like 0.611 there, and similar trends are observed in in the AUPR side. But I think that's actually a message I want to deliver to the community. It's like, yeah, I have to speed up. I'm almost done. Yeah, it's like, yeah, so it's like still this is a challenge. It's like if you have one cell line information, it's still hard to predict for the other cell line. And I also compare my methods to all the existing methods out there using a very good reference data sets thanks to the ELISO paper. And I just want to share that it's there are multiple. The community are trying really hard, but all these data sets are like thousands of samples levels. It's not a big data set, but I was able to show that my method is always the top 2 performer there in a purely data-driven way compared to using references from Knowledge Graph or PPI or Go. And that's the summary of my project. It's like our methods are good, but still have a lot of space for improvement, especially if you look at the PR curves. There are a few points. Firstly, it's like basically my features, the distribution of my features are still not covering the whole synthetic lethal base space enough to predict it, although it's like 4 million pairs there. And actually the way we define synthetic lethal now is still vague because we literally just make a single cutoff in the scores saying that, yeah, this part is synthetic lethal, that part is non-synthetic lethal. So this is not a very consistent gold standard there. And it really depends on the cell line context and your data set quality. And if we really have this computational model, we don't want to just stop at comparing in the benchmark statistic. We really want to use it to guide the new screens. So we have to quantitatively evaluate how efficiently it really drives the new screens compared with you just randomly pick the next candidate to do the experiments. And as what I see from the cell line generalization problem is that that's a big motivation towards some foundation way. Actually, it's like transfer learning or like pre-training. It's like if you have all the cell line information in your training process and it's large enough to cover all the space, maybe you can train something like a big autoencoder or transformer-based architecture to collect all the cell line information and then fine-tune on each cell line context. That would be an ideal venue for my next exploration. So with that, I would like to just thank everybody from our lab and our collaborators, and thank you for listening. Any questions?
Hi, thank you so much for such an insightful presentation. I just wanted to ask, in the context of getting these drug targets, as in the synthetic lethality GIs, how does your method take into accountability the healthy tissues interactions that could become toxic in these healthy tissues? Because you only touched like the cancer side of things. So I'm thinking usually the gene interactions differ from tissue to tissue. So was there any, or how did your method take into accountability such? Thank you.
Yeah, thanks a lot for the question. So I think, yeah, so we do have to find more specific cell line context dependent interactions or cell states dependent interactions. It's like, we have to make sure it's not really affecting the healthy cells. And even if the mutations only happen in a certain cell types, we don't want that interactions to impact some rewiring processes. So more factors could be considered, including cell states, tumor mutation environment, transcription factors, things. Those are more things that we can look at in the future. But currently we are like this experimental in vitro status, and that will kind of expand into like a higher tissue level of like tissue or in vivo models. So that's a long way to go. But at least we want to provide this data set to show the community that, yeah, these are interesting pairs you can start looking at. Because as I show, it's like many of these pairs are even not screened before. So if you don't screen it, you don't know what effect it really has, and you don't know the potential target there. So it's a part of this is a big experimental efforts, and part of my ambition is to computationally infer the rest of them so that people can start looking at them even before they are screened. So that's the goal there. Thanks a lot for the question. Yeah.